Today we're going to discuss RNA-Seq, which is another way of looking at a large set of genes. So as we talked about last week, this is another way to look at possibly thousands of genes and then filter it down to a few genes that you're interested in pursuing further based on your research question. So before we jump into RNA-Seq, I want to do a quick review of the nucleic acids in the cell. As you all know, there are 4 bases that form the backbone of all the nucleic acids in the cell. These bases are adenine, thymine, cytosine, and guanine. They are classified as purines and pyrimidines, and a good way to remember that cytosine and thymine are your pyrimidines is to remember this acronym CUT1Pi, which tells you they have 1 ring and they're pyrimidines. These 4 bases come together to form our double-stranded DNA, which is basically the genetic code, and it is the same in every single cell. So every cell is going to have the same DNA, the same exact structure, and the same exact amount of DNA. This DNA is going to be transcribed into mRNA. mRNA stands for messenger RNA, and this is the RNA that is actually carrying out which genes need to be transcribed and which genes are going to be expressed. So although all of the DNA is the exact same in every single cell, as we talked about, the mRNA is going to change based on which genes are being expressed within the cell. So not all DNA is going to be transcribed to mRNA, even though all of the DNA is present in every single cell. This mRNA is then going to travel out of the nucleus. So if we think about this as our nuclear boundary, it's going to travel out of that, and then it is going to be translated into protein. Now the standard code is that all of the DNA gets transcribed into mRNA, which gets translated into protein. But we know that not all the DNA is transcribed. It's specific based on what the cell wants to express at given points in time. So the mRNA is changing based on the conditions of the cells. And we also know now that not all mRNA is translated onto protein. Sometimes mRNA just gets degraded, sometimes it isn't translated and it's kept around for later needs of the cell. So it's important to realize that the mRNA and the protein levels will not always correlate in every single cell. But for the most part, the canonical idea is that the mRNA does go out of the nucleus and then is translated to protein. And this process is something most biology classes will cover. Here we're not going to cover it in a great amount of detail. Basically, every 3 bases are going to code for 1 amino acid here, and together they're going to form a chain of amino acids, which will then fold into proteins, which then perform functions within the cell. The protein is what actually does everything within the cell, so it is important to also understand the levels of the proteins, because that is what is actually making a difference. However, looking at levels of mRNA can give us a pretty good approximation of what is going on at the protein level. However, as we've discussed, sometimes it gets degraded, sometimes it isn't translated, so it's not a perfect one-to-one scenario. The other thing we can do with mRNA is reverse engineer it to become cDNA. cDNA stands for complementary DNA, and it is essentially a backbone version of the mRNA. So it's taking the mRNA and it's going back 1 step and saying, what is the DNA that this mRNA was transcribed from? And this is important because sometimes it's important to have DNA for downstream reactions. So when we're doing sequencing, when we're doing PCR, when we're working with certain applications, we want to have DNA and not mRNA. And so we take the mRNA and we make DNA out of it. But this is not real DNA because it's not the DNA that's in the cell. It's only the DNA that was transcribed to make mRNA. So it only represents the DNA that was being turned on to express certain genes. It does not represent all the DNA in the cell. This is useful, as I said, because sometimes we need it for specific applications, and so it's something we do in the lab fairly commonly, so we can use cDNA for further downstream things. So just to take a little bit of a closer look at making cDNA from mRNA, because as we discussed, this is a very common lab reaction because we want to use it for downstream applications. A very common example of this is PCR. So the way this reaction works is you have a tube, and this tube has your mRNA in it. So this is your template that you're making the cDNA from. Into this tube, you're going to add reverse transcriptase, which is a special enzyme that can generate DNA from an RNA template. You're going to add primers, and these are just primers that will help the DNA form. So for DNA to form, if this is your mRNA strand, DNA cannot form without a little bit of a primer here. It needs something to build on. So these primers just provide a backbone for the DNA to build on. It also has DNTPs. These are just your bases. So these are A, T, G, and C. It has some water, and it has the buffer with all the necessary inhibitors to ensure that the reaction actually does proceed and doesn't get contaminated by anything. So then looking at the actual reaction, if you imagine that this black is your mRNA strand right here, this green thing is the primer, so this will come and anneal at random places along the mRNA in order to give the DNA a start point. The reverse transcriptase, so this here is your reverse transcriptase, will then come and attach to this primer and this mRNA strand and will generate the cDNA that you see here, which is the yellow. So this is our cDNA that's slowly growing along the strand. And then once that part of the reaction is over and the cDNA has been generated, you heat up the reaction, which will inactivate the reverse transcriptase, and you have your final strand of cDNA generated. This allows us, like I said, to make DNA from mRNA with the correct bases, because remember that mRNA uses a U instead of the T. So this allows us to have actual DNA bases, so then we can do other things in the lab that we may need to do with the mRNA or with the transcript. Okay, so now with that background, let's get into RNA-Seq itself. So we're going to start with a basic overview of what RNA-Seq is, how it came to be, and what some other methods are that we might use that are comparable to RNA-Seq. So first we'll discuss what RNA-Seq measures. So RNA-Seq measures what's called the transcriptome, and the transcriptome is just a fancy word for saying all the genes that are actually being transcribed. So as we've discussed before, the cell has all the DNA. Every single cell has the same DNA, but what makes different cells do different things is which genes are actually being transcribed. And so we can talk about the genome, and that's just all of the genes. But then we can also talk about the transcriptome, and that's going to be all the genes that are actually turned on at any one time. And so RNA-Seq is really looking specifically at the genes that are being transcribed. And it does it through NGS, which is next generation sequencing, which we'll get into in just a second. But first, as a reminder, we know that this is our DNA. DNA gets formed into mRNA, and this is what RNA-Seq is measuring. And this is how we know that when we do an RNA-Seq, we're looking at what is actually being expressed in the cell. If we wanted to go back and make cDNA out of this, as we just discussed, the cDNA strand would look like this bottom strand here, because this is what the RNA was transcribed off of. And then this mRNA is further translated to protein, which is what this is here. And there are other ways to look at this, but RNA-Seq looks specifically at this mRNA piece right here. So now we understand what RNA-Seq is measuring. Let's look at how it does it. So this is a quick overview of next generation sequencing. It's sort of very broad. There's a lot of details in NGS, but a lot of the time companies will do this for you, so you don't need to know all of the details. You need to have a basic overview of what's going on. So the way NGS works is you send a library. This library should be everything that you want to sequence. So this is what you're sending for sequencing. Usually it's genomic DNA, or it can be some form of DNA that you've generated. So in our case, it might be cDNA that we've generated from mRNA. So this is our mRNA. We then fragment this mRNA into pieces so that the cDNA can handle it. So the sequencer can handle it a little bit better. So it gets fragmented, and then it gets adapters added to it. So these adapters are just so the sequencer can recognize which sample this is coming from, and also recognize that this is something that needs to be sequenced. And then you ligate it together, and this forms your library. So this is everything you're sending to sequencing. When you send for sequencing, you're going to amplify what needs to be sequenced. This just allows for you to have enough that your sequencing is going to work properly. After that, you're going to perform the actual sequencing. And so the sequencing cycles occur, and the data is outputted to a text file. It basically makes reads for each small fragment, because remember, we have made fragments and we've amplified those fragments, and now we have multiple fragments, and we're reading each fragment one by one. And then once all the fragments have been read, so imagine these are all of our different fragments that have been read, but these are all fragments that come from the same sort of long sequence that we want to know. So this is the sequence that we actually want to know, and these are all the fragments that we made from that sequence when we cut it up before. So if we now, if we remove these cuts just so we can see the sequence, these are all the fragments. The sequencer has sequenced each fragment, so it has a sequence for every single fragment. And now it's going to align these fragments together in order to figure out what the actual sequence should be. So it matches up multiple fragments and generates the actual sequence of the whole entire piece before fragmentation. And so now this is your sequence, and this is what you can move forward with for analysis. So let's just take one more look at the sequence alignment and how it works to make it very clear. So these are original, let's say these are original mRNAs, which you've converted into cDNAs. So these are original DNA pieces that you want to sequence. To send them for sequencing, you're going to chop them up so the sequencer can handle them a little better. So now you have all these chopped up fragments that you're sending for sequencing. And the sequencer will spit out a sequence for every single one. So for example, for this, it might be like, okay, the sequence is ACGT, whatever. And for this one, the sequence might be CGT, ATT, and so on. And now it's going to take these fragments and it's going to sort of use all the different fragments to together align them and figure out a picture of what the original sequence must have been. And then this is the sequence that it actually gives you at the end. Okay, so having discussed a little bit about RNA-Seq and how it measures the mRNA level, and having also discussed how that actually works and how the sequencing works, I just want to take a minute to step back and talk about how new RNA-Seq is and how interesting and cutting edge of a tool it can be. So just to give some perspective, 1869 was when we first discovered the DNA molecule even existed. We weren't able to figure out the structure until 1953. And then 1961, we figured out the codon. And 1967, we figured out sort of a very basic DNA sequencing, nothing like what we have today. It was 1983 when we finally figured out how to do PCR, which is really key for any sort of amplification and any sort of downstream application of DNA. And once we had that, we were able to develop the microarray, which we will discuss next. And that's sort of the old version of RNA-Seq. In 2000, we developed next generation sequencing, so that's only 20 years old. And it wasn't until 2008 that it actually became a usable tool. So in fact, it's really only been 12 years of being able to use NGS, and we've already done so many cool things with it. So NGS is what allows us to do RNA-Seq, but it's also what allows us to do ChIP-seq, CRISPR-Cas9 screening. There's a lot of different tools that now depend on this high throughput sequencing that we're able to do. And so as soon as we had next-gen sequencing, we were then able to develop RNA-Seq. And so then in 2010, you start seeing publications based on RNA-Seq data. So this whole technique is only 10 years old. And we've already done a lot with it, but that really highlights how new it is and how much potential it still has. If we look at this table of publications, you kind of see the same thing. Everybody used to use microarrays, so those became a thing around 2000. And there were lots of publications with those. But then RNA-Seq became a thing at 2010, and you can see that now that's really going to start taking over how we do these experiments. And if we look at this since 2010, when RNA-Seq started, becoming widely used, and that's what this is, there's also all of these other different sequencing methods that we've started doing. And so it's really given us a lot of power to do a lot of different types of sequencing and a lot of different types of applications. And now there's much, much more that we can do with this technique beyond what we're going to talk about today. So this is just really here to highlight, how new this is, and also how much other new stuff we're developing and how much potential there is in the field. Okay, so I mentioned microarrays on the previous slide, and I do want to go through these in some detail because even though they are the legacy experiment and we don't do them as much anymore, you will still see them in lots and lots of papers, because pre-2010, this was the most widely used way to measure gene expression, and so it's important to understand how it works and how to interpret it. So as an example, let's say that we have a culture of normal cells, and we're interested in comparing this culture to a culture of cancer cells, because we want to know what is the difference between the genes that the cancer cell expresses and the genes that the normal cell expresses. And remember that this comparison is going to be at the mRNA level, as we discussed before. So also, like we discussed before, you'll create cDNA from your mRNA using the reaction we talked about. And when you do this reaction, you're going to add red fluorescence to your cDNA for the cancer cell, and you're going to add green fluorescence to cDNA for the normal cell. And then your microarray chip already has specific cDNA probes on it. So this is important because you have to already know the genes you're looking for. You cannot use a microarray to look for unknown genes because you have to have a cDNA probe already on the chip for the gene that you're interested in looking at. So for a number of known genes, we can look at the expression at the mRNA level and compare it between a normal cell and a cancer cell. And when we do that, we're basically looking at the red versus the green. So if we have more green, it means the normal cell expresses it more. And if we have more red, it means that the cancer cell expresses it more. So for example, this red one here means that the cancer cell is expressing this gene more. And this green one here means the normal cell is expressing it more. If we have yellow, it's present in both cells. And if we have gray, that means we see no color, so there was nothing attaching to that probe. So the gene is not expressed in either cell. So just to go back through the interpretation one more time, in this case, our sample of interest is going to be green. So this is the patient sample. And our reference sample or control sample is going to be red. So this is an example of hybridizing against a sample of reference and then looking at a sample of interest, which would be the patient one. So we mix these samples and we hybridize them onto this chip. And the way the chip works is, if this is your chip, you can imagine that every single circle has a single strand of DNA for that gene. So this is a cDNA for the gene. But this is complementary to whatever is going to be in your sample. So if your sample has that cDNA, then it's going to come and it's going to go ahead and bind, and so you'll get the color. But if your sample doesn't have the cDNA, then you're not going to see any binding, and so then you're going to see a gray result because there's not going to be any binding. In this case, since we have a reference which has everything, you'll always see binding, but you'll see red in those cases, and so you'll know that that gene is not as highly present in your sample of interest. So samples are along the top, genes are along the side, and here we can kind of see all of the different possibilities. So if we put all of our different samples along the top, and then these are all of the genes that are available, we can kind of see how the gene is expressed in every single sample. And then this gets run through the computer, and that analyzes what is gained and what is lost. So it's telling you which genes are elevated based on your microarray and which genes are reduced based on your microarray. So if we take the computational version of this, if you imagine that you have a yellow spot, that means that you have the same number of patient samples to control. So you have a 1-to-1 ratio, which means there's no difference in expression. If you have a green spot, that means that you have more of your green samples, you have more of your patient samples. That means that their gene is up in the patient as compared to your reference. And if you have a red dot, that means that your gene is going to be down compared to your reference sample. So if we translate this to a table format, then you can imagine that if these are all of your different genes here, this is your full change, which is how green or red it is. This might be the actual absolute value of what you're seeing for each sample. And this would be the p-value, which would be how significant that change is. This is what we actually see. This is sample microarray data. And this is a sample of what the microarray might look like. So to go through the difference between RNA-Seq and a microarray, just to review, RNA-Seq is going to be based on sequencing, specifically next-gen sequencing. So because we're sequencing exactly what every single transcript is individually, we can see whether there are novel transcripts, and we can also have really, really high specificity because we're literally going through and sequencing every single transcript that is available. And so as of now, this is the preferred method to look at gene expression if you want to look at thousands of genes or a large array of genes. Microarray is kind of the old method that was used very widely, and it's really based on this matching of having one DNA strand already on your chip and matching the complementary DNA strand to it, which would come from your sample. And that has that fluorophore attached. So for example, the red or the green fluorophore. And this is really good for identifying known transcripts. So you have to know what gene you're interested in to be able to have this probe, right? If you don't know the gene, if it's a novel transcript, then you can't have a probe, and so you wouldn't be able to identify it. And because it has to be known and because it's based on matching, it also has lower specificity because you're not sequencing every single transcript and getting a raw count. You're looking at colors and the different amount of each color. So you can have some noise, especially for genes that are low expressed. And so it's harder to figure out the actual expression level of those genes using this method. So this is really considered a legacy method at this point, but like I said, important to understand because it is in a lot of papers. Okay, so now that we fully understand RNA-Seq and we understand microarrays, let's just talk very briefly about what kind of research question RNA-Seq answers. So our last video focused on CRISPR-Cas9, and in that we talked about how CRISPR-Cas9 is really functional questions. So you are doing a knockout or a knock-in of every single gene in the genome, and you're screening across all of those knockouts. And so that's really a question of what is the function of this gene? If I knock it out, how does this function change? And therefore, what can I infer about the function of this gene? And it allows us to look at phenotypes or functions across a large number of genes. RNA-Seq, on the other hand, is really more about expression. So it's not going to tell you anything about the function, although you might say that, if this gene is expressed in this scenario or under this condition, then perhaps it has this function. But it doesn't directly answer a functional question. It's a lot more about what is the expression change or what genes or pathways are turned on after I expose the cells to this condition or after this amount of time or after this treatment. So it's really just answering purely what's up and what's down after a certain condition. So it's important to understand that distinction because based on what you're trying to understand, you might choose a different screening type. So far we've talked about these two, but there are also screens that help you look at proteins or help you look at epigenetic modifications that we'll discuss in the future. So thinking about which screen is appropriate for the question you're asking is an important step in deciding what to choose. So now that we've gone through RNA-Seq and the general theory of how it works, let's talk about how we actually use RNA-Seq in the lab. So this is more about how do you practically set up an experiment and how do you execute it and analyze it and how do you interpret all the results. So let's look at the basic setup. So when you set up an RNA-Seq experiment, your first thing is to choose which model you're going to do this experiment in. So you can choose to do an RNA-Seq across multiple cells. You can also choose to do RNA-Seq with an animal model. You can also even do human samples if you are able to get human samples. So you have a lot of choices. Once you choose the model that you're interested in using, you also need to choose the conditions. So RNA-Seq can allow you to compare 2 or more than 2 conditions. It's just important to keep in mind what the appropriate control should be. So a simple version might be that I want to do a control and a treatment. So I want to compare control cells and cells treated with TMZ or some other drug. A more complicated version might be that I want to look at primary human samples and recurrent human samples. So this is a time change, and hopefully you can get matched samples from the same patient so that your control is appropriate. So your control in that case would be the primary, and you would be comparing against the recurrent. So the important thing is just to make sure that your control is the best control possible and the closest to your drug condition or to your treatment condition. You can do these over time as well. So you could say my control is day 0 and my treatment condition or experimental condition is seeing how the cells are acting at day 10 or day 20 or any time point that you're interested in. And you can combine these designs. So you could say that I want to do a control and a TMZ experiment at day 1 and day 4 and day 8 because I want to know how the cells change both with addition of the drug and over time. I want to see if there is a difference between the reaction to the cells at day 1 of TMZ and day 8 of TMZ, which I might expect that there is. And so doing this RNA-Seq setup allows me to do that. But if I were doing that, I would have to make sure that I had the appropriate control, which would be a DMSO control, at day 1 and another one at day 4 and another one at day 8 so that I have the perfect matched control for every single drug time point. That way the only thing I'm changing is the drug. I'm not also changing time. If this was the only control I had, then I would be changing time and drug with these TMZ samples. So just make sure that your controls are very well matched. So once you have your appropriate controls and treatments, then you're going to extract mRNA. And from this mRNA, you're going to generate cDNA, which is what we've discussed before. And then you're going to send that for sequencing once you have that cDNA prepared. The only other thing to keep in mind is that ideally these controls and treatment conditions, whatever you choose to do, should be in triplicate. So you're going to have a minimum of 6 samples if you send this experiment. And if I sent this experiment, then I would actually have 18 samples. The triplicate allows you to really make sure that your results are reliable, and so it's good practice to do triplicates at all points if you can. So once we send it for sequencing, it goes through the sequencer. This, for almost all labs, is going to be a black box. You don't really need to worry about exactly what's happening during sequencing. We've talked about a basic overview of how the sequencing works, and that's really all you need to know. You basically send your cDNA into the sequencing, and you're going to get out this FASTQ file. And this is where it will come back to you, and now you need to think about what to do again. So a FASTQ file is going to be all of the sequences of the cDNA fragments that you sent in. And once you get this FASTQ file, it is important to check the quality. So this quality check will allow you to check how well the sequencing happened. So how reliable are the sequences that you got out, and were there any issues in the sequencing? So this is an important first step to make sure that your sequencing was good. And if it wasn't, then maybe you need to send the sample back to the company or ask them to do it one more time to get a better sequencing quality. So once we have this FASTQ file, which looks like this, we're going to align these sequences. So when we align these sequences, you remember that we sent a bunch of cDNAs to the sequencing core. So these cDNAs are all going to align to the genome in a specific place. They're all going to be mapped to a specific gene on our genome. So for example, this might be a gene called blue, this might be a gene called orange, and this might be a gene called green. So these are 3 genes in our genome, and we have taken the sequences we got and mapped them back to this place on the genome. And now, based on what we're seeing here, we're going to generate a table of counts. So if I were generating this exactly off of what I'm seeing here, this would be 6, this would be 2, and this would be 4, which says that I have 6 copies of this blue gene that I sent for sequencing, and 2 of this orange, and 4 of the green. So this is basically saying that I have, these are all of the things that I sent for sequencing, and this is, once I map them, what I actually see in the genome, how many copies of each one I have. So this example is just an example of a count table. So once you put in all of your different replicates, so let's say we sent control and treatment, and we sent 3 replicates of each one, then these are going to be all of our controls, and these are going to be all of our treatments. We're going to do this FASTQ and alignment with each one of these samples, and we're going to get a certain number for each one based on how many copies of that gene are found in the sequencing files. So now we've generated a count table that says, for example, in your control sample for the first replicate, there were 10 copies of gene green that were seen in the sequencing file, and there were 37 copies seen in your control sample, your second control sample for the orange gene. So this just summarizes all of the different gene counts that were seen, okay? Which is the same thing as saying that it summarizes the cDNA levels in every sample that you sent. Okay, so this is going back to this and basically counting exactly what did you send by analyzing the sequencing files and mapping them back to the genome. This is another place where it is really important to check the quality. Now that you have your counts, you want to make sure that the counts make sense. There's no crazy outliers, and that all of your samples look similar. So if you send 3 replicates of a sample, that all of the replicates look similar and make sense. And then once you have this count table and you've checked the quality and you know that all of your samples look good and are appropriately similar, you can go ahead and do an expression analysis. And so this expression analysis we'll talk about in more detail, but it commonly uses an algorithm called DESeq2, which allows you to take this count table and generate a score of how much things have changed, so a fold change, and a p-value, which tells you how significant that change is. And then these are 2 ways of visualizing that we'll get into later. So I just want to note that this pipeline that we've gone through is basically the same with some minor modifications for all sequencing applications. So whether you send a CRISPR or an RNA-Seq, a ChIP-seq, whatever you're sending, you're always going to get back a FASTQ file. You're always going to need to check the quality. You're always going to need to align to a genome. And you're going to get some sort of count table that tells you, for CRISPR, it might be guides. So how many copies of every single guide do you have? For RNA-Seq, it's genes. How many copies of every gene? For ChIP-seq, it might be how many modifications do you have of every single area on the genome? But it's always a count table that has what you actually need. And then you analyze that count table using some sort of algorithm, and that gives you a score and a p-value, which is what you use to decide what is actually important. So if you understand this for one application, it's fairly easy to extend it to other applications. And this time we're going to spend a lot of time walking through this pipeline so that we can understand it really well and extend it to other things. So let's start by looking at a FASTQ file. So this is exactly what comes out of the sequencer. When you send something for sequencing, this is the first type of file that is going to be spit out. And this contains everything that you need to know about the sequencing. So it contains the actual sequence information. It also contains a lot of quality information. It gives you IDs and lengths of all your sequences. It gives you confidence scores. So it's really quite rich with information. So this is the information you get for one sequence, these first 4 lines. And this is the information you get for a second sequence. So for each sequence, I get an identifier, which is just a tag that identifies that specific sequence. And it also gives me a length. I get the actual sequence, so this is what was actually sequenced. This is just a sign saying the strandedness. And then I get quality scores. So this gives me, these are letters and they're defined by Illumina to tell you how good the quality is of every single base. So it basically tells you how confident is the sequencer that this base is actually a T or that this base is actually a G. And this is what is used to determine the quality of the sequencing. So there's another algorithm that takes in these strings and tells you how good is the quality of your overall sequence based on what it's seeing here. So let's take a look at that quality analysis. So this is assessing quality of your FASTQ sequencing file. So this is just sequence quality. It's not telling you anything about your counts or your analysis. It's only telling you, is this sequencing file a file that you can trust and analyze further, or is it a file that needs to go back? So these are all the things that FastQC gives you. So we're going to go through a few of them. This is your basic stats. So this tells you what your file is, and it gives you an idea of how many sequences were in it and how many were flagged as being poor quality, what the average length of your sequence is. So this is some basic information about your sequencing file. This is your per base sequencing quality. So we said that we have 40 base sequences, so this goes from 1 to 40. So it's going across every single base. And it's basically saying that at base position 1, how good is the quality of my sequence? So is it 36, which is the highest quality, or is it going down to 0 or 2, which is the lowest quality? So across every single sequence in my entire experiment, how many of them have a sequence that's 38? And if almost all of them do, then it's going to be up here in this green area. But if a lot of them are 24, then I'm going to see the histogram bars extend down to 24, and so on. So for example, if these bars extended really far down, then I would know that at the 3 end of my sequences, there were a lot of mistakes. But the way it looks right now, we know that overall the bases look really good, and our sequence quality looks really good across the entire length of the sequence. This is a similar plot here, except that it summarizes all of the sequences together. So this one gives you a per base analysis. So it's saying base 1 versus base 5 versus base 32. What does every single base look like? Whereas this is just looking at overall, how many sequences had an overall quality score of over 36. So it's saying that 16,000 of my sequences, or more than 16,000, had quality scores that ranged in the really high end. So that tells me that my sequence quality looks really good. This one is showing me per base sequence content. So that means if I have reliable sequencing, I should basically have the same concentration of T's versus C's versus G's versus A's, because we know that there are only 4 bases in the DNA and they show up at reliable percentages. So if I take every single sequence and I look at base 1, there should be an equal percentage of each base pair in that region. And so basically you should just see consistent lines across the board, which is what we see here. And finally, there is a graph for adapter contents. This tells you if your sequencing data has been contaminated by any known adapters. So adapters are often added into sequencing while the sequencing is happening to allow the sequencer to recognize the sequences and amplify them. But they're always removed during the sequencing, or ideally they should be removed during the sequencing so they don't interfere with further analysis of your FASTQ files. So this just shows you whether you have any adapter contamination or not. And in this case, since everything is 0, we do not have any adapter contamination. So having looked at a good example, let's take a quick look at a bad FASTQ example. So here we're looking at per base sequence quality, like we just did, and we have all of our bases going from the 5 end to the 3 end. And you can see that at the 3 end, we have a lot of bars that are extending all the way down to 2. So 2 is a bad quality score, 34 is a good quality score. So what we're seeing here is that at the 3 end, we have a lot of mistakes. There is very low confidence in the quality of this sequencing. The sequencer is telling us that it's not sure what these bases are. And so if there are this many mistakes, we're extending this far into the red, that like half of our sequence is in the red zone, then this really isn't sequencing data we can trust, because we don't know whether half of the sequence is correct. And so it's really hard to align this to a genome and actually trust it. So this is definitely a sequence that should go back. If we look at our per sequence quality, we see the same thing, that there's a bump around 17. So 17 is halfway, right? So that tells us that, again, there's a big bump in low quality sequences. And then we also have quite a few high quality sequences, but having this many low quality sequences makes it hard for us to trust the data. When we look at our per base sequence content, instead of seeing those nice straight lines that we saw last time, we're seeing a lot of jumping around, which also doesn't make sense, because when we look at thousands of sequences, we would expect that there would be consistent percentages of all of the different base pairs, because we know that they're consistently present in the DNA. And so the fact that it's all jumping around and jumbled up is not convincing. We need it really to be those straight lines that we saw before. And then this one looks at adapter content. And so red is the Illumina universal adapter. And we can see here that it is contaminating the sequence. As we go further along the read, this is our position in the read. And we saw before that it should have ended at 40, but now we're seeing that we have these reads that are much longer than 40. And these reads are highly contaminated with the adapter. So basically the adapter, if this is our actual read, the adapter has just been tacked on to the end. And this is going to make it really difficult for us to align this to any genome, because it's not going to match the genome. So there are ways to just cut this out from the FASTQ file, but sometimes it's better to just go back and get a cleaner file that you can then use for further analysis. So once we've looked at the quality of our FASTQ files and we know that they're good quality files, we can move on to the alignment. So far we've gotten our FASTQ files. We've sent stuff to the sequencer. From the sequencer, we've gotten the FASTQ files. And now we've checked the quality. Quality is good. And now we're going to align the files. So aligning the files means that we take all of the fragments that we sequence, that we now know are good quality, and we match them to where they should be on the genome. And when we match them to where they should be on the genome, it allows us to figure out what gene this is. So for example, maybe this blue gene is LNX1. And so now we know that we have 6 copies of LNX1 because we matched it to the genome, and this is the genome location of LNX1. So this alignment is really important to help you figure out the count table, because only once you align, you can figure out how many copies you have and what gene it corresponds to. There are many programs that allow you to align. A lot of them are in Python, and so for now, think of it as a black box. A lot of companies, when they do sequencing for you, will send you the alignment, and there are a lot of online tools available, so it's not really something you need to worry about just yet. Just make sure you understand exactly what is going on, that it is being aligned and matched to a genome. And then from that genome matching, we're able to generate a table that says this is the gene of interest and this is how many copies of that gene you have in each sample. And like we said before, these are replicates of control and these are replicates of treatment that we're looking at. And then the final thing to note, even though you don't really need to worry about exactly how to align, it's good to know that the latest human genome is HD38. So this is what the latest experiment should be aligned to and will give you the most correct count tables. Okay, so now we've done our alignment. So we've gone from sequencer, we've made our FASTQ files, we've aligned our FASTQ files, and we have a count table. And now that we have a count table, we really want to check and make sure that the samples are similar. So what that means is that replicates should be similar and conditions of interest should be different, because we're trying to figure out what the difference is between our conditions. So if there's not really too much difference between the conditions, then all we've learned is that there's not that much difference between the conditions and there's nothing much to say about it. So for the quality assessment, it's important to look at exactly how similar or different your samples and replicates are. There are 2 ways to do this. One is a clustering heat map, and the second is called a principal component analysis, or a PCA analysis. Both of these depend on the same thing. They basically take a measure of distance between each replicate or sample that you're interested in. And this, again, you can think of it as a black box for now. It can be done in R. There are many different algorithms that allow you to do it fairly easily. And R can generate these figures for you, again, fairly easily. But for now, let's just focus on understanding what these figures mean. So let's start by looking at the clustering heat map. So these are our conditions. So let's say N is one condition and T is another condition. We have 4 replicates for each, and then we have the same thing. We have a T here and an N here. The blue and the red is a measure of similarity. So the more blue something is, the closer it is to 0, and the more red it is, the closer it is to 1. So we would expect that our replicates would cluster around either 0 or 1, because they should be similar to each other. Replicates should be similar to each other, but they should be different from the condition. So if N and T are 2 conditions, then within N, we're seeing a lot of blue, which tells us that the N's are all similar to each other. But between N and T, we're seeing blue versus red, which tells us that our conditions are different from each other. So you're basically just looking at clustering of colors. And you want to see similar colors within replicates. So similar colors within replicates, and different colors between conditions. And that is what tells you that your conditions are different, but your replicates are similar to each other. The other way to look at this is to do the principal components analysis. So for this analysis, you're basically looking at how they cluster in 2 dimensions. This first dimension is the dimension where they show the greatest difference. So this is sort of one measure of difference between the 2. And the second dimension is where they show the greatest measure of difference, the second greatest measure of difference. So this is going to be the second most important way in which they are different. And so focusing on just the first most important way in which they're different, which would be this axis, we see that all the reds cluster on one side and all the blues cluster on the other side. So red is our control and blue is our treatment. And so we can see that the first most important thing that separates the 2 of them, or the thing that makes the most difference, is another way to think about it, because we're seeing 51% variance. So the thing that makes the most difference is that they are treated. But then the thing that makes the second most difference, if we look at this, is that they are different cell lines. So if we look at the shapes, we can see that we're using 3 different cell lines here. And so these 2 cell lines are fairly similar, but this cell line is different, and it's different for both of them. So you can see that the thing that second most makes a difference is that this cell line, whatever it is somewhat different from the other 2 cell lines. But if you're looking at this plot, you would still say that the most important thing that makes a difference between 2 samples is the treatment. The treatment is creating a change that is a much bigger change than the change created by the fact that there are different cell lines involved. If the samples were actually truly similar, so if these were 3 replicates of the exact same sample and we didn't have this cell line difference, then I would expect to see 3 red dots right here and perhaps 3 blue dots right here, which would show me that they're different in 2 dimensions, but they're entirely similar to each other because they're in the same place on the plot from each other. So this tells you difference in one dimension versus difference in another. So it can give you sort of 2 measures of difference. This clustering can give you, in a way, only 1 measure of difference because you're really just looking at color change. But PCIs can be done in 2, 3, 4, 5 dimensions, and you can really see that do these cluster across all of the different dimensions, or are there things that create change? If I look at multiple dimensions, do I see that there are changes depending on the dimension I look at? So this is useful. This is useful in general because it creates a nice visualization. And if I had perfect replicates, I might see visualizations like this. but it's also useful when you have multiple conditions you're looking at. So if you're looking at untreated and treated and also cell lines and also time, then perhaps you would want to do 3 PCI plots so you could really see which condition makes the most difference and how these separate in space. So right now what this is telling me, just to recap, is that the greatest difference is created by the treatment, because that is the greatest variance, but that there is some variance introduced by the fact that there are different cell lines and that this cell line is different than these 2 cell lines. Okay, so now that we've assessed all of the different components, so to repeat, we've gone from our sequencing, we've made our FASTQ files for which we've checked the quality, we've aligned them and generated count tables, and we've checked the quality of those as well. And now we're ready to take our count tables and run the actual algorithm that tells us what has changed. So what has changed and how significant is that change? And the algorithm for this is known as DESeq2. This is something used fairly commonly for all kinds of gene analyses, and so it's useful to understand not just for RNA-Seq, but for other applications as well. So what DESeq2 is doing is it's taking in your count table, it's running it through, usually it can be run in R, it's running it through DESeq2 with some sort of design formula. So your design formula is going to tell it what is the change that I've made. So in this case, my design might be just treatment, and so that tells DESeq2 that I have a control and I have a treatment. If I had, for example, a time change as well, then I might add treatment plus time, and that would tell it I have control and treatment at 2 different time points. And then it takes this design that you give it, and it generates an analysis table, which gives you a score and a p-value. And from this analysis table, you can create visualizations of the data and show which genes have changed the most. So just to take a closer look at what DESeq2 is actually doing, you can imagine that it follows this workflow right here. So it's going to take in your read counts. This is basically our counts table. It's going to normalize those counts, and then it's going to perform the actual measurement of what the change is. So to do this, it sort of estimates how different every gene is compared to the mean, and then it shrinks those differences and fits them to generate a score and a p-value. This is a very well-validated algorithm, so you don't need to worry too much about the details. You can trust that it's going to generate a reliable score and a reliable p-value, and so you can focus on these log2 fold changes and thinking about which genes have actually changed. But just to give a very simple overview of what it's doing, if you imagine that you have 2 samples, you have condition A and condition B. Blue is going to be our condition A, and red is going to be condition B. These pink circles are going to be the mean for each group, and the yellow is going to be the global mean. And so what DESeq2 is doing is it's just comparing this distance and determining if it is a great enough difference to be significant or not. So in this one, there's a significant difference between this mean and the global mean. But in this one, for example, all of the molecules are jumbled together. And so if we look at our 2 global means and we look at your yellow circle, they're all almost on top of each other. And so we're not going to see a significant difference between these 2 groups. So let's say that you did want to run DESeq2 on your own. Let's say that you were able to get this FASTQ file and you were able to get the counts table, and now you just want to analyze it. So there are a few ways to do this. One is this app called the Green app, which was very recently published. This is useful because there are a number of RNA-Seq datasets that have already been published, but they haven't been very accessible because people don't always know how to run the analysis. So there is now this app that allows you to search for interesting data that has been published, but might be interesting as a comparison to your own data or might provide an interesting correlate to your paper. And it provides the DESeq2 analysis, so you can just look at full changes and scores and include that information in your paper as long as you cite it. The other thing that's useful is DEApp, which will take in count tables and generate all of the analysis for you. And with this, you can use your own data sets. So this is a useful tool for analyzing DESeq2 data, even if you didn't know how to code and you just wanted to be able to do an analysis. You can also write it in R, and it is fairly simple to write in R. So this is a pipeline that I like, that I followed along with, but there are many pipelines available that allow you to write it in R. So I'm just going to go over a very simple script that allows you to write this in R. So the first thing you have to do is import the DESeq2 package. You have to import it from Bioconductor, so you import it exactly like this. And then you have to load it into your workspace. So this library command will load the package into your workspace so that you can actually use it. Then you're going to set the working directory. So you can think of the working directory as what R is actually seeing. So these are the files that it can access and that it can see and that it can play with. And so you are going to set this to wherever your file of interest is. So this is going to be ideally wherever your counts file is, so that R can see the counts file and pull it in. So whatever path that is. And then you're going to import the data files. So you're going to say counts, and you're going to use this command to import that counts table that you have as an Excel file or a CSV file into R, so that R can see it and analyze the data and work with the data. This command then allows you to create a DESeq data set. So R has a specific type of data frame that's called a DESeq data set, and it allows you to do DESeq functions. So it allows you to do specific plots, specific analyses, and it has all the necessary metadata and control information that DESeq needs to do the analysis correctly. So when you take your counts and you give it a design, you can generate this object, and this object is what allows you to do everything moving forward. Once you have the object, use these two commands to actually perform the DESeq2 calculations. So this will perform the entire pipeline that we discussed on the previous slide. And this results is what's going to contain your actual results. So you can see that now that we want to export it, I've saved the results information to a variable called results, and then I've written it out to a CSV file. So this will take you from a count table to the DESeq2 analysis. There are, of course, ways to do all of the quality analyses and the plots, the PCA plots, Volcano plots, heat maps as well, but this is just the simplest version that gives you that Excel file that has all of the DESeq2 statistics that you would need. So now let's understand this results table. So this was the table we put in. This was our counts. And we ran DESeq2 on it, and we got a results table. So this table has a few different things. So the first thing it has is the gene. This is just going to be the name of your genes. Okay. Then you're going to have a base mean. The base mean is going to be the mean of all of the counts for the sample. So this includes any treatments and controls all together. You're going to have a full change. So this full change will always be calculated as your treatment condition over your control condition. So this is going to tell you, is my gene up or is it down compared between my samples? It's going to give you a standard error. So this is, how confident am I in this log2 fold change? What is the error of this log2 fold change? It's going to be a statistic, and this statistic, you can think of it kind of like a t-test statistic or, any sort of statistical test statistic. It just allows you to figure out the necessary p-value. So this is usually something you don't have to pay too much attention to. What you do want to pay attention to is the p-value, because a p-value of under 0.05 denotes significance. So that's how you can figure out whether your gene is changing significantly over time or not. And then DESeq2 also gives you an adjusted p-value, which is actually probably better to use because it's been adjusted appropriately for your sample design. So the two things you're going to pay the most attention to usually in these tables is the log2 fold change and the adjusted p-value. And this is going to give you, did my gene change? And if it did change, how much did it change? And how significant was that change? So once we have that table of results, we can do two visualizations with it. One is a heat map, which we're going to go through here. So this right here is a sample heat map, and we'll go through this step by step. So let's say that we have 3 samples for each condition. So this is plain GBM cells, this is GBM with a drug, and this is GBM with vitamin C. We have 3 replicates for each sample. And the first thing we're going to look at is how well do these replicates cluster. So what we should see is that each of these clusters together, and that's what we kind of see with these lines here. They tell us that, you know, if we started at a common ancestor, these split, and then this became 1 cluster, and then these split again, and these became 2 clusters. So this one is the least similar because they've split off the first, and then these 2 are slightly more similar to each other than they are to this, but they're still different from each other because they split again to form these 2 conditions. So now that we know that all of our samples are clustered appropriately, so the replicates are clustered together based on the map we're seeing up here, and they're different from each other also based on the map we're seeing up here, we are comfortable analyzing these replicates because we know that the replicates are appropriate. So then we're going to look at our full change legend. So the full change legend here tells you what every color means. So here, a blue means that there's a -4 full change, which means the gene is down by 4 units, or log 2 4 units. And if it's red, then it's up by 4 units. So this is important to look at because the colors can change from heat map to heat map, and so you want to make sure that you're interpreting it correctly. Our genes are going to be along our y-axis here, and again, they're going to be clustered by similarity. So the clusters tell you, is the pattern of change similar across this set of genes as compared to another set of genes? And then every single box that we're seeing here is going to represent the full change. So if we're looking at this box, it would be the full change of this gene, and it would be in this sample. And then we could compare that by color and see that it's becoming more blue over time, so this gene is getting down-regulated over time. The other way to look at the gene changes is to look at a volcano plot visualization. So this plot is going to look at p-values on the y-axis. This is going to be the log 10 of the p-value, and it's going to look at the log 2 fold change on the x-axis. So this is going to be upregulated genes, and this is going to be downregulated genes. This is going to be less significant and more significant. Usually you will set a significance cutoff. So this line might be, for example, p is less than 0.05. And so everything above it, because this is a negative log scale, everything above it is going to be significant, and everything below it is going to be not significant. And then you actually plot the genes. And usually if you have a good analysis, then you'll see this volcano plot shape where it's sort of a parabolic shape. And so the genes that come above our significance cutoff and below the full change cutoff are going to be considered downregulated genes because they have a lower full change, but they are significant. And the ones that go above our log full change cutoffs, they have a positive log 2 full change, are going to be upregulated genes because, again, they're significant, but they're above the cutoff, so they're upregulated. And if we're looking at targets that we're interested in, what we really want to look at is targets that are significant, extremely significant, but also that have changed a lot. Because if we have something that has a big log 2 fold change, like 2 to the 5th is telling us that there's a 32 fold change, for example, for this gene over our control condition, that means that this is a gene that's changing quite significantly, and it's something we might want to look at in our actual lab and sort of validate at the bench. And so usually it's the things that sort of fall out of the analysis that come towards the end, these outliers that are very significant and also highly changed, that end up being the things people want to look at. So these right here, or any of these right here, would be genes that would be interesting to look at. So today we've gone over RNA-Seq and microarrays. We've discussed the basics of how RNA-Seq works and how to interpret it. We've walked through the analysis step by step. each piece of the pipeline and how that works. And we've also talked about how to interpret key RNA-seq visualizations. If you have any further questions about this video, please feel free to contact me here. Thank you so much for listening.